Hello and welcome to another edition of Zog Science. Today we're going to be talking about cancer and a cool type of biotechnology called DNA chips or DNA microarrays. So, what is cancer? Cancer is a disease that occurs when a cell loses control of mitosis. Remember, mitosis is the process that occurs in just your normal everyday body cells or somatic cells that goes where is the process of cell division, how your body replaces damaged cells or cells that have died. And cancer is when that process of mitosis somehow becomes abnormal and your cells begin to divide uncontrollably. So they're basically going through mitosis over and over and over again without slowing down at all. And what that does is it creates a mass of cells, which is known as a tumor. Just kind of some characteristics of normal and cancer cells. Normal cells tend to be a little bit more symmetrical or a little bit more circular in shape, very, very clean looking, whereas your cancer cells typically a little bit abnormal. They can sometimes have multiple nuclei inside. You have lots of nucleoli, as well as the chromatin is going to look very coarse and very much darker as opposed to your normal cells. So again, we kind of talked about this. Tumors are just groups of cells that begin to divide uncontrollably, so it's not just a single cell, but it's the whole group. And tumors are what generally cause damage when it comes to cancer because they can take over different parts of your body. This right here is a liver that is very cancerous, and you can see all these white places. This is all the different tumors, and the tumors take nutrients away from the other tissues or different cells that need them, as well as can actually push things out of the way and press on different parts of your body, causing pain or loss of function. And when a cancer cells kind of break loose, so let's say some cancer cells broke off of here and spread to another part of the body, say the liver or the kidneys, that is not good, and that is when you have a malignant tumor. It's also called metastasizing. One of the most important genes, although it's certainly not the only gene, is the p53 gene, and it codes for a protein that halts the cell cycle until all the chromosomes have been properly replicated. So it basically ensures that no mutations or the fewest number of mutations have occurred. And if you have a damaged or missing p53 gene, this can lead to a lot of issues and a lot of different types of cancer. It is one of the most strongly correlated genes in terms of seeing it not being expressed or it missing, and also seeing cancer production. There are lots of different other genes that are involved in different cancers. There's the BRCA1 and 2 genes that are involved in breast cancer. But again, most of the time cancer is both a combination of environmental and genetic factors, and it's also generally a multifactorial, multi-gene genetic issue where you can have multiple genes that are all working together in order to form cancer. So how does this kind of relate to DNA chips or microarrays? DNA chips are very powerful technology, allow us to scientists see what genes are being expressed in a cell. And if we can see what genes are being expressed in a cell, then we can investigate whether or not what genes are being expressed when a cell is cancerous and what genes are being expressed when a cell is normal. So how does this all work? Well, basically we take a microscope slide and we coat it with all of the DNA pieces for a gene, and we actually put the non-coding sequences of DNA on the microarray. And the reason why we do that is what we're going to do is we're going to take the mRNA that we find in a cell. So basically we take a cell, we get the mRNA that's in it, and if we find the mRNA, then that must mean that the gene is being expressed, because remember it goes DNA, mRNA, protein. So we find the mRNA that is in a cell, we take that mRNA and we turn it into DNA. So we actually work backwards and we use an enzyme called mRNA reverse transcriptase. So we take the mRNA, we turn it into DNA. Then what we do is if we want to compare a normal and a cancer cell, we will label all the mRNA from the normal cell with this lovely green color and all the mRNA from the cancerous cell with the red color. We are then going to take those pieces of cDNA, is what they're called, complementary DNA, and we are going to hybridize, or basically wash them over the slide. And if the pairs are complementary, because remember we put the non-coding DNA over here, whereas this is the cDNA is also going to be the same as the coding DNA, they're going to hybridize or complement each other and bind. And that means that they're going to stick onto the plate. What we then do is we take a laser and we scan over them, and whenever the laser hits the red ones, it's going to show up red, and whenever it hits the green ones, it's going to show up green. And if you have both of them, then that means that it is yellow. So I've also got a lovely little animation. These are just kind of the steps. If you want to pause the video and write them down, you can. But I'm just kind of going to move forward to the animation. So here we go. So here we've got our two cells, all right? And what we're doing is we are adding in some extraction buffer. So now we've got the mRNA kind of just floating around in the liquid. We're then going to take that out using our lovely micropipettes, which are obviously we used in the gel electrophoresis lab. They're pretty wonderful. And obviously we want to get a new one. So we're going to take that mRNA and put it into a fresh tube so that we only have the mRNA. So now we're going to make the cDNA, or that complementary DNA. So here's our mRNA molecules. And we're going to make one of them green and one of them red. So we use this lovely colored DNA, and really it's not colored, it's just that when a laser hits it, it's going to reflect light. The cDNA is made, and then we use a process to get rid of the mRNA, so that all we have left is the cDNA. And notice that we have them in our separate tubes, so we're going to put them together, so that way we can then put them onto the same slide. Alright, so we've got them together. And next we're going to take a look at our DNA chip and wash them over. So here's our chip. Each spot is going to represent a different gene, right? And so we're going to take those, and usually there's going to be a lot more than what was shown right there. But what we do is we have our base pairs, right, for the genes, whichever one it is. And we're going to take that mixed DNA, the one that we had before, and we are going to put that over top of our DNA chip. And what's going to happen is that, remember, they're going to complement, and if they complement, then that means that the DNA is going to stick and stay, which, remember, it has this nice color. All right. So now we can wash off all of the stuff that did not bind, so any pieces of the cDNA that did not bind. And we're just going to take a look at these three spots, and this is where we shine our lasers over the top. So the green laser is going to come along and find out where we have the green DNA. Then the red laser comes along and looks at the red DNA. And now we're going to basically just look at our computer results. So it knows exactly where it had the green ones and where it had the red ones, and it's going to merge the two together. So what this means is that the green ones are, in this case, this particular experiment was looking at whether or not it was anaerobic or an aerobic or anaerobic gene. But we could also say, you know, this gene is expressed in a normal cell. This gene is expressed in both a normal and a cancer cell, and this gene is expressed in only a cancer cell. So which are the two genes that we actually want to investigate? Well, it's going to be these two right here, because these ones are the ones that are changing based on whether or not it is a normal cell or a cancer cell. So we would investigate both of those genes, figure out what they do, and then kind of proceed from there. So why do we want to use microarrays? Okay, well, they're very powerful, and they allow us to examine hundreds of genes at once. We don't have to just kind of look at one at a time. We can do all of them. It's a very easy way to determine which genes are being expressed or repressed, which is the opposite of being expressed, in different types of cells. So maybe a cancer cell is repressing a particular gene or expressing it. And again, it's just very useful in terms of looking at, you know, in diseased agents, whether or not there are particular genes that are being expressed or not expressed. All right, that's the end of the notes. We will be doing a lab where we're going to take a look at DNA chips, so look forward to that, and I will see you all next time.